For decades, biological and medical communities viewed aging as a process of stochastic entropy. Stochastic entropy is a statistical concept describing the random, inevitable accumulation of wear and tear that leads to the failure of a system. Under this model, human aging was seen as a passive, irreversible breakdown. Recent research has replaced this view with a biological model that defines aging as a highly regulated process. Scientists now identify aging as a series of specific molecular and cellular mechanisms that are both predictable and targetable. To organize these mechanisms, researchers established the Hallmarks of Aging framework. In 2013, the field identified 9 foundational hallmarks. By 2025, that model expanded to 14, incorporating systemic determinants like the gut microbiome and psychosocial factors. To be classified as a true hallmark, a biological process must meet 3 strict criteria. It must manifest predictably during normal aging. Its experimental aggravation must accelerate the aging phenotype, and its experimental attenuation must decelerate the rate of aging, extending the healthy lifespan of the organism. This framework demonstrates that aging is not a linear timeline of decay. It is a self-amplifying biological network, where a single failure in one node propagates across the entire system. To systemize the 14-node network, geroscience categorizes the hallmarks into a tripartite causal hierarchy. This tiered structure defines exactly when and how each biological failure emerges. At the base are the primary hallmarks. These are the negative root causes that initiate the aging cascade by accumulating irreparable biochemical lesions at the cellular level. The middle tier contains the antagonistic hallmarks. These represent compensatory responses that protect the body from acute damage in youth, but become toxic and pro-aging when they are chronically hyperactivated in later life. The peak consists of the integrative hallmarks. These are the systemic consequences that emerge when the body's defenses fail to contain primary damage, leading to the clinical collapse of tissues and organs. Precision geromedicine uses this hierarchy to determine its strategy. Clinicians aim to target the primary root lesions at the base to prevent the systemic failures at the top. The cascade begins with the unrelenting accumulation of primary damage. This includes genomic instability, where double-strand DNA breaks go unmasked, and telomere attrition, the progressive shortening of the protective caps on our chromosomes. This damage triggers epigenetic alterations, which lead to transcriptomic drift. Transcriptomic drift is the progressive loss of the precise gene regulation required to maintain a cell's identity, resulting in genes being transcribed inappropriately. The result is cellular confusion. A cell loses its function and erroneously reactivates ancient silenced retroviruses within its genome, triggering internal inflammation and genetic chaos. The 2023 update elevated disabled macrophagy to a primary hallmark. Macroautophagy is the cell's essential recycling program that breaks down and reuses damaged organelles and large protein aggregates. This failure is marked by the accumulation of cytosolic LC3 that fails to undergo lipidation. Lipidation is the process of attaching a lipid molecule to a protein. In this case, it is required to convert LC3 into LC3-2, the form needed to clear cellular waste. When transcriptomic drift occurs and cellular recycling shuts down, the cell is left cluttered with broken, active machinery. This internal crisis activates Tier 2 compensations. Tier 2 antagonistic mechanisms are biphasic. While they protect the organism during reproductive years, they become the primary drivers of pathology in old age. This is seen in deregulated nutrient sensing. The mTOR kinase complex, which senses amino acid abundance, becomes chronically hyperactivated. mTOR is a protein complex that regulates cell growth, and its overactivity tricks the body into staying in a pro-growth state even as its infrastructure fails. This chronic mTOR signaling actively suppresses macroautophagy, directly linking Tier 2 hyperactivation back to the Tier 1 failure of cellular recycling. Without recycling, tissues accumulate damaged mitochondria that produce less energy and leak massive amounts of ROS. ROS, or reactive oxygen species, are unstable molecules that inflict oxidative damage on DNA and lipids. To prevent these damaged cells from becoming cancerous, the body triggers cellular senescence. This permanent cell cycle arrest acts as a frontline tumor suppressor. However, these senescent cells resist death and develop the SASP. The SASP, or senescence-associated secretory phenotype, is a continuous secretion of pro-inflammatory cytokines and enzymes that poison the surrounding tissue. The massive continuous secretion of this SASP cocktail transforms a localized protective arrest into a spreading toxic environment that accelerates aging across the entire organ. In Tier 3, the accumulation of SASP and ROS damage overwhelms the body's homeostatic resilience, the ability of an organism to maintain internal stability, leading to tissue collapse. This manifests as stem cell exhaustion. The toxic signals from nearby senescent cells corrupt the stem cell niche, destroying the body's capacity to regenerate tissues or heal wounds. The failure extends to the microbiome, resulting in dysbiosis. This is the loss of beneficial eubionts and the rise of pathobionts. Eubionts are healthy gut bacteria that maintain the intestinal barrier, while pathobionts are microorganisms that can cause disease when the ecosystem is unbalanced. This imbalance makes the gut leaky, allowing LPS to translocate into the blood. LPS, or lipopolysaccharides, are toxins from the cell walls of certain bacteria that continuously stimulate the immune system's toll-like receptors. This continuous endotoxemia, combined with SASP secretions, creates inflammaging. This unresolved, low-grade systemic inflammation acts as a primary catalyst for all late-stage age-related diseases. These three tiers do not operate in isolation. They form a self-amplifying feedback loop. Metabolic imbalances like mTOR hyperactivation inhibit recycling, which causes mitochondrial ROS leakage. This damage inflicts further DNA damage, triggering senescence, which secretes SASP and ultimately exhausts systemic stem cell pools. External factors can accelerate this molecular loop. Psychosocial isolation chronically activates the HPA axis, the body's central stress response system, elevating cortisol levels and driving further systemic inflammation. Because aging is a connected network, intervening at precise nodes can sever these pathological loops and halt the progression towards systemic collapse. Precision geromedicine is the clinical application of this framework. It moves away from treating late-stage symptoms to target the underlying molecular determinants of the aging network. One strategy targets nutrient sensing using rapalogs. These are drugs that inhibit the mTORC1 complex, forcing cells out of a pro-growth state and upregulating the macroautophagy recycling program. The TAME trial, targeting aging with metformin, aims to activate the AMPK sensor. By doing so, researchers hope to convince the FDA to grant aging its first official therapeutic indication, recognizing it as a treatable clinical condition. Clinical data has also revealed geroprotective effects in GLP-1 receptor agonists. These drugs, originally for diabetes, effectively dampen systemic inflammation by modulating nutrient-sensing pathways. To clear senescent cells, clinicians use senolytics. These drugs transiently disable the anti-apoptotic survival pathways, like BCL2, that these cells rely on to survive, causing the zombie cells to finally undergo programmed cell death. By pharmacologically clearing the senescent burden and resetting metabolic sensors, clinicians can mute the toxic signals propagating through the antagonistic tier of the hierarchy. The most ambitious frontier in the field targets the primary hallmarks directly through partial cellular reprogramming. This technique attempts to reverse the root epigenetic alterations that drive the aging cascade. This process uses the Yamanaka factors, 4 specific proteins that can erase the epigenetic marks of age from adult cells, effectively resetting their biological clock. However, this therapy must be transient and partial. If the factors are expressed for too long, cells lose their functional identity and can form teratomas, which are tumors made of a chaotic mix of different tissue types. When combined with microbiome engineering and senolytics, these interventions aim to restore the hallmarks of health. This means returning the body to a state of robust repair and internal stability. The 14-node network model shows that aging is not a mystery of inevitable decay. By mapping and modulating these biological determinants, medicine is moving aging from an unmanageable decline into a tractable biological variable.